Hello and welcome to another episode of Katie the Science Lady. I'm Mrs. Jacobson, and today's topic is the first part of protein synthesis, transcription. So let's learn together. Okay, today we're going to talk about the first part of protein synthesis called transcription. So we know that protein synthesis is a process where we take DNA and then we eventually turn it into a protein. Because DNA, that holds our genetic material, our genetic instructions on how to make every protein in the body. But we have to first solve a problem. We've got a problem here. We need to use the material we have in the nucleus, our DNA, but we can't use it to make a protein until we get it to a ribosome, which is located in the cytoplasm. DNA can't leave the nucleus. So we've got our instructions stored as DNA in the nucleus. Please remember that one gene is equal to one protein. So one section of DNA is equal to one protein. Now our ribosomes, again, are outside in the cytoplasm. DNA cannot leave. It's too big. This is designed on purpose. If DNA could leave our nucleus, it could get damaged out in the cytoplasm somewhere. And we only have one copy of our DNA in each cell. So if we damage it, it's damaged for that cell forever, unless we can repair it. So we don't want our DNA to leave the nucleus because it's dangerous. So instead, we have to make a messenger. We have to create a messenger molecule that's a copy of DNA that can also leave and go to the cytoplasm. And our solution is messenger RNA, also called mRNA. Now let's kind of talk about the differences between DNA and RNA quickly. DNA is deoxyribonucleic acid. It has the sugar deoxyribose, which is that first part of this name. It's double-stranded. That's why we call it the double helix. It's got that kind of twisting spiral staircase structure. The bases are G, C, A, and T. Remember, guanine, cytosine, adenine, and thymine. RNA, on the other hand, is ribonucleic acid. So the sugar is going to be called ribose. It's single-stranded. That's why it's skinnier. It can fit out of the nuclear pores. It's just smaller. And the bases are G, C, A, and U, or guanine, cytosine, adenine, and uracil. So that's a key difference here. Uracil is going to replace RNA, where we would have thymine normally. So we've got two different bases here. We've got thymine, which is DNA, but it's no longer there. And in RNA, it's now uracil. Back when we talked about DNA structure, I kind of mentioned the base pairing rules, and we had apple in the tree, A pairs with T, car in the garage, C pairs with G. So we still have those base pairing rules, except the car still parks in the garage, so G still pairs with C, but now the apple is under the tree. So I kind of think of it that way. Apple's underneath now. Like, it's different. The apple's fallen. It's under the tree. That just helps me remember where it gets paired. So if we're looking at this DNA strand here and we're making an mRNA copy, it's going to read, so again, the T here, T pairs with A, C pairs with G, A pairs with, we have a U over here, and I know you can't see it, and we'd have, again, a C would pair with a G over here. So we've got our new strand over here for our mRNA. It's single-stranded. We have R's here on the sugar for ribose. There's still a phosphate. So instead of being a sugar, phosphate, and a base, it's still a sugar, phosphate, and a base, but it just happens to be ribose, a phosphate, and then one of our four bases, A, G, C, and U. So back to transcription. We're making this copy. It's essentially just a messenger. It's a message. That's why we call it messenger RNA. It's just a copy of this recipe for a protein, and we're sending it out of the nucleus. mRNA is smaller. It's only single-stranded, so it's half the width, and it can leave the nucleus. So we see that in here. When we're making it, there's an enzyme called RNA polymerase, and its job is to just build mRNA. It reads the DNA strand and decides which of the nucleotides need to be added to make mRNA, that copy. As soon as that's finished, mRNA leaves the nucleus and goes to find a ribosome. I want to go back through this process one more time here. It's pretty simple. There's not a super crazy process here. It is more complicated than I will be going through, but that's more of an AP Bio level. So if you need any of those resources, please go ahead and find other videos on YouTube to kind of help solidify your learning. So when we look at our DNA, that's going to be in blue here. It's been opened with a helicase. Remember, helicase is one of our other enzymes that's going to unzip our DNA, just like we talked about with DNA replication. Now it's going to unzip this DNA and allow the RNA polymerase, which is this really big enzyme here, that kind of grayish enzyme shape. It's going to allow that to add RNA nucleotides, the A's, the U's, the G's, and the C's, to that first strand. We call it a template strand, but it's going to break off. It doesn't stay connected to that template strand of DNA. It makes a single strand using the RNA polymerase, and then it leaves. So just going to use that as its template, and then it leaves the nucleus. So if we are going to change or go through transcription, here's what it would look like. In our DNA, we have like AT, A, G, A, T, C. Normally, if we're making DNA pairs, this would be a T. It would be AT as a pair. But now we're making mRNA, and mRNA, there aren't any thymines. There are no T's. So we have to change every T we would see into a U. So instead we have A pairs with U. T, we can still have DNA T's, that's fine. They pair with an A. A pairs with U again. G still pairs with C. A pairs with U because it's mRNA. T pairs with A. And C pairs with G. The most important thing to remember is if you have a line here of mRNA, there should be 0 thymines in it, none. You cannot have them, they don't exist. mRNA bases, thymine just isn't one, so you're not going to see it. Here's another example. If we have CCG, AATT, it will pair up this way. C pairs with G, C pairs with G, G pairs with C, A pairs with U, A pairs with U, and then T's pair with A. This is just something that takes a while to practice. Anytime there should be a T, you want to make sure that it's a U because mRNA cannot have a T. That's a good way to check your work. So after you go back through and say T pairs with A, G pairs with C, C pairs with G, if you say A pairs with T and you look at this line and go, oops, nope, that can't be a T. It has to be a uracil. So just keep that in mind. Anytime you're writing mRNA or you're reading mRNA, there cannot be any T's, no thymines present. Here's a quick GIF of this process. So helicase is unzipping the DNA. We've got this big molecule here is our RNA polymerase. And what it's doing is it's pairing up according to the base pair rules. Now it's pulling in those A's, U's, C's, and G's. As soon as it pairs up, it starts leaving that RNA polymerase. It's using words like terminator. So as soon as it gets to the end section of the gene, it's going to stop. And once transcription stops, that mRNA is going to leave the nucleus. And you can see it floating here in this GIF. So we've got that floating mRNA. Let's review what we've learned so far about protein synthesis and about transcription. Protein synthesis starts off as a problem. We need to get out into the cytoplasm to use ribosomes to make proteins, but the DNA we use is in the nucleus and it can't leave. So we need a messenger molecule to take this DNA message into the cytoplasm to the ribosome in order to make our protein. That's where transcription comes in. Transcription is the process of taking DNA and using RNA polymerase to make a messenger copy of this DNA called messenger RNA or mRNA. To make mRNA, RNA polymerase matches up the base pairs G and C, guanine and cytosine, and A and U, adenine and uracil. There is no thymine when we're making mRNA. This mRNA molecule is single-stranded and can be leaving the nucleus through pores in the surface. Once the copy leaves the nucleus, it travels to a ribosome in the cytoplasm and is ready to start translation, the second half of protein synthesis. Well, that's it for today. Thank you very much for watching. Please like, subscribe, and comment to my channel. I enjoy having you here. And until next time, I hope you had fun. I hope you learned something, and I'll see you later.